Hello everyone, welcome back to this educational channel called Learn Bio with Janet. Today we will discuss meiosis, part 2, the second video on meiosis. In the first video we have already defined what meiosis is and where it occurs and also the importance of meiosis. Today we'll study the process of meiosis, and that is meiosis 1 and 2. So this topic is from the Form 4 KSSM syllabus, Chapter 6, Subtopic 6.3, and the chapter is entitled Cell Division. The learning outcomes for this lesson are as follows. Firstly, after the lesson, we should be able to explain the stages of meiosis in the correct order. That is, we are going to discuss the stages of meiosis 1 and the events that occur in the different stages. And this is followed by meiosis 2. Secondly, we must be able to draw and label the cell structure in each stage of meiosis 1, meiosis 2, and then cytokinesis. And thirdly, we will compare and contrast meiosis 1 and meiosis 2. Let us first revise the definition of meiosis. Now, meiosis is a type of nuclear division or division of the nucleus that occurs in reproductive organs such as the ovary and testes to produce gametes. Gametes are the reproductive cells involved in fertilization and formation of the new offspring. Now, these gametes contain half the number of chromosomes. That means they are in haploid condition of the parent cells, which are in diploid condition. So haploid condition means that the cell or the gametes have only one set of chromosomes, and the parent cells are in diploid condition, meaning that they have two sets of chromosomes. So a diploid parent cell produces 4 haploid daughter cells, which are gametes, such as sperm, ovum, and pollen. So referring to this picture, we start off in meiosis with the diploid parent cell having 2 sets of chromosomes, or there are 2 copies of each type of chromosome. Then in meiosis 1, the homologous chromosomes will separate and then the cell becomes haploid. N stands for haploid. That means having only one set of chromosomes. Okay, compared to the diploid parent cell, which has 2 sets of chromosomes in it. Then after meiosis 1, the nuclear division, there is cytokinesis or cytoplasmic division. That's how the cell splits into 2. Then next, these daughter cells will undergo meiosis 2, where the sister chromatids will separate and move to different poles, and then cytoplasmic division occurs to produce 4 daughter cells in all, and they are haploid. Okay, and some of them are genetically different from each other and from the parent cell. So how this occurs will be discussed further on. Let us discuss the meaning of the terms paternal and maternal chromosomes and homologous chromosomes before we discuss meiosis. Now, in a diploid cell, there are 2 sets of chromosomes. One set of paternal chromosomes originate or come from the male parent or the father. It can be denoted by the red color chromosomes. Okay, this is one set of paternal chromosomes, all of different sizes, carrying different genes. And B, one set of maternal chromosomes, which originate from the female parent or the mother, denoted by the yellow colored chromosomes. So mother has one set of chromosomes, also of different sizes, carrying different genes. So because there are 2 sets of chromosomes, one set of paternal chromosome, one set of maternal chromosome, thus for each type of chromosome, we have 2 copies. One copy is the paternal chromosome and one copy is the maternal chromosome. Okay, so this forms the pair of homologous chromosomes. Forms a pair of homologous chromosomes. Now, what are homologous chromosomes? A pair of paternal and maternal chromosomes which have the same structural characteristics. For example, same length, same position of centromere, and same type of genes. The only difference is the version of the genes may be different. Okay, so let's read here. However, homologous chromosomes may carry different versions of a gene. For example, let's look at this long chromosome, medium-sized chromosome. Maybe here in the paternal chromosome, there is a gene for blood group O, but mother may contribute the gene for blood group A here. So the version of the gene for blood group is different. And here maybe you have father has a gene for black hair, but mother may have a gene for brown hair. Okay, so different versions of the gene. So this means that in the pair of homologous chromosomes, the two chromosomes are not genetically identical. The paternal chromosome and the maternal chromosome are definitely not genetically 100% identical. Okay, so one copy of the homologous chromosomes is from the father and one is from the mother. Here is an overview of the stages of meiosis and the other events that occur before it and after it. So meiosis consists of two separate nuclear divisions, that is meiosis I and meiosis II. Now in meiosis I, there are four phases: prophase I, metaphase I, ANAPHASE I, and TELOPHASE I. So we can use the acronym PMAT1, PMAT, PMAT1, to remember the four phases of meiosis I. And briefly, prophase I, in prophase I, synapses and crossing over occur. So we will be discussing these two processes later. And then in ANAPHASE I, homologous chromosomes will separate and move to different poles of the cell. And finally, the daughter cells formed will be haploid. From diploid condition, they will become haploid after meiosis I. Now then, meiosis II consists of, again, four phases: prophase II, metaphase II, ANAPHASE II, and TELOPHASE II. So it's PMAT2. Then in ANAPHASE II, sister chromatids separate. That's the important event in meiosis II. Right. Now, before meiosis occurs, first there must be replication of DNA during the S phase of interphase. Okay, then meiosis I occurs. After meiosis I, the cell will undergo cytoplasmic division, which is called cytokinesis. So after meiosis I or nuclear division, then you have cytokinesis or cytoplasmic division. Then only we have interphase, a short interphase, and then the cell goes into meiosis II. Then after meiosis II, or at the end of meiosis II, there is another occurrence of cytokinesis. So the two daughter cells formed after meiosis I will then divide again and form four daughter cells in the end. So here's a picture to help us understand a bit more about the overview of meiosis and the events that occur before and after it and during meiosis. Now at the beginning in interphase of the cell cycle, in the G1 phase, if we take a cell with only a pair of homologous chromosomes, at the beginning the chromosomes are unduplicated. So these two chromosomes are a pair of homologous chromosomes. One is the paternal chromosome, the other is the maternal chromosome. So they are not duplicated yet. And in here, in this diploid cell, where there are two copies of each type of chromosome, the number of chromosomes is 2. So 2n equals to 2. 2n is the number of chromosomes in the diploid cell, and there are 2 chromosomes here. Now, then in S phase of interphase, duplication of chromosomes occur and the DNA replicates. So now each chromosome has duplicated and it consists of a pair of sister chromatids which are genetically identical and they are attached together at the centromere. Okay, so the number of chromosomes is still the same. This is considered one chromosome and this is considered another chromosome. So there are still two chromosomes, only two chromosomes in the cell, but the DNA mass has doubled compared to the original cell here. Okay, DNA mass here is 2 times the DNA mass here. Now, next, in meiosis 1, during anaphase 1, the homologous chromosomes separate from each other and move to different poles. And then finally, there is cytokinesis or division of the cytoplasm. So then only one chromosome, one of the chromosomes will be found in each daughter cell form. So that means there's only one copy of this type of chromosome in each daughter cell. It's considered one copy of the chromosome. And this is the haploid condition. So from diploid condition, where there are two copies of this type of chromosome, now there's only one copy of this type of chromosome of this length. So we call this a haploid daughter cell. So from diploid condition, the cell has become haploid after meiosis 1. Then in meiosis 2, sister chromatids will separate. So these two sister chromatids will separate from each other when the centromere divides. And the sister chromatid will be pulled to the opposite poles. The two sister chromatids will be pulled to the opposite poles and they will separate. So each sister chromatid will become a new chromosome in each daughter cell formed. The cell will divide again during cytokinesis to form 2 daughter cells each. So in the end, we have 4 daughter cells formed, which are haploid. So here are some notes related to what I said just now. In cell division involving meiosis, a cell undergoes 2 nuclear divisions, meiosis 1 and meiosis 2, 2 cytoplasmic divisions, that is after meiosis 1 and another time after meiosis 2. That's how we get 4 daughter cells from one diploid parent cell. And C, replication of DNA or duplication of chromosomes occur only once during S phase of interphase before meiosis. To summarize what we have discussed in the previous diagram, here is the overview in words. The overview of meiosis. Meiosis 1 begins with one diploid cell. In prophase 1, 2 processes occur which are very important. Synapsis and crossing over occurs that causes genetic variation or differences in organisms. We will discuss this later. In anaphase 1, homologous chromosomes separate from one another and move to opposite poles. So at the end of meiosis 1, cytoplasmic division has also occurred after the nuclear division. So 2 haploid daughter cells are formed. In meiosis 2, the cell begins with 2 haploid daughter cells. But this is the intermediate stage. So in anaphase 2, sister chromatids in each chromosome separate from one another and move to opposite poles. The sister chromatids will separate. And at the end of meiosis 2, 4 haploid daughter cells are formed. The 4 daughter cells, which are the gametes, are genetically different from each other and from the parent cell. We will go into the stages of meiosis 1 and 2 so that what we have discussed here will become clearer. Let us now discuss what happens in meiosis. We will start with meiosis 1, and the first phase of meiosis 1 is called prophase 1. In prophase 1, some events that occur are similar to those in prophase of mitosis. For example, The chromatin will shorten and thicken to form visible chromosomes, which are in the shape of an X. And each chromosome is seen to consist of two sister chromatids. For example, one sister chromatid here and one sister chromatid here, attached together at the centromere. Okay, two sister chromatids attached together at the centromere. Next, two events occur which do not occur in prophase of mitosis, but only occur in prophase I, okay, of meiosis. The two events that occur in prophase I of meiosis, which are very important, are synapsis and crossing over. Now, in synapsis, homologous chromosomes will come together. For example, the pair of long chromosomes will come together and pair up to form bivalence. So here we see the two copies of the homologous chromosomes, one paternal chromosome and one maternal chromosome, pairing up. So this pair of chromosomes is called a bivalent. Bi meaning two, so two homologous chromosomes together, pairing up together. Or they are also known as a tetrad. Tetra means four, because there are four chromatids together. Alright, so either bivalent or tetrad. So next, during crossing over, the next stage, non-identical chromatids, meaning a chromatid from the paternal chromosome and a chromatid from the maternal chromosome. They are non-identical. They are genetically non-identical because, as we said, paternal chromosome may carry genes that have different versions. They may carry different versions of genes compared to the maternal chromosome. Father may have gene for blood group O, mother may have gene for blood group A. Father may have gene for brown hair here, and mother may have gene for black hair. Okay, so they are not identical, genetically not identical. So these non-identical chromatids or non-sister chromatids of homologous chromosomes may exchange genetic material. So when they exchange genetic material, they will touch each other. The chromatids will touch each other to exchange genetic material. And you can see the process occurring here. Now this is called crossing over. Alright, so crossing over is important because it allows exchange of genetic material between paternal and maternal chromosomes, and it will produce new combinations of genes on the chromosome, where the genes on a paternal chromosome will combine with genes from maternal chromosomes. So the point where they cross over here is called the chiasma, where they touch each other. The plural form is chiasmata. Let's find out more about crossing over. Let us discuss more about crossing over, which is very important for the exam. It's a popular question. And crossing over is one of the most important events that occur in meiosis. Alright? So in prophase I, first of all, there's synapsis, where homologous chromosomes come together and pair up. Next, they undergo crossing over, where there's exchange of genetic material or segments of DNA between homologous chromosomes. One chromosome is the paternal chromosome, and it exchanges genes with the maternal chromosome. Okay, so at the point where they touch, where the chromatids, non-identical chromatids touch, that means chromatid from the paternal chromosome touch with the chromatid from the maternal chromosome. Then at the point where they touch, that means the chiasma here, they will break off and join to the other chromatid. So at the end of crossing over, we find that this chromatid here has joined to a segment of the maternal chromosome has joined to the chromatid here. And here the paternal chromosome, a segment of it has joined to the maternal chromosome. Okay, so at the end of meiosis, the sister chromatids will separate from each other, move to opposite poles, and they'll be found in different daughter cells. Same for these two. Sister chromatids will separate and move to opposite poles, and they'll be found in different daughter cells. So here we see 4 daughter cells formed from this is from one parent cell. And these are called the gametes, and they are genetically different from each other because they have different combinations of genes. For example, this chromosome has the paternal genes from the father of this individual that is producing the gametes. And this gamete has the maternal genes. But these two in the center here, they have new combinations of paternal and maternal genes. Alright, so gametes produced will be genetically different, although they are produced from the same individual. And if these gametes undergo fertilization, they'll produce genetically different offspring. So this will cause variation in the population, in the species. Now, what is the importance of variation? First of all, variation are differences in characteristics between individuals of the same species. For example, differences in the blood type, the color of the hair, the height, and also differences in resistance to disease. So it's very important to have differences in resistance to disease. We'll discuss further about the importance of variation in the next slide. Here is an important question that is commonly asked about meiosis. Explain an important process that occurs in prophase I of meiosis, or you may be given a diagram like this and asked to explain the process that occurs here, which is crossing over. Not crossover, crossing over. So let's see how to explain the process and its importance. So in prophase I, homologous chromosomes undergo crossing over. The exchange of genetic material or segments of DNA occurs between non-identical chromatids, that means between the paternal chromosome and the maternal chromosome, producing new combinations of genes, new combinations of paternal and maternal genes. Now, this produces genetically different gametes from one individual. So an individual will produce gametes that are mostly genetically different, leading to variation. So if these gametes undergo fertilization, they'll produce offspring that are genetically different, and this causes variation, which are differences in characteristics between individuals of the same species. Now, what is the importance of variation? Variation increases the ability of a species to adapt to changes in the environment. For example, if a disease were to spread among in the human population, some people with weak or low resistance to that disease may be struck down and may be affected and may fall sick. But for others, because they have different resistance to disease due to variation and due to the fact that they are genetically different, so these individuals may be able to overcome the disease and may not even fall sick, for example, because of the different resistance to diseases. So in that way, it will help the species to survive. It will help humans to survive because some of the members or some of the members of the species will survive due to variation. So the following are the events which occur in prophase I. I'll just go through very quickly. Chromatin shorten, thicken, and form visible chromosomes. Each chromosome consists of two sister chromatids attached at the centromere. In synapsis, homologous chromosomes come together and pair up to form bivalents, also known as a tetrad, that is, 4 chromatids are found in each pair of homologous chromosomes. Now, in a bivalent, one of the chromosomes is a paternal chromosome and the other is a maternal chromosome. Next, during crossing over, non-identical chromatids, that means the non-sister chromatids of homologous chromosomes, exchange genetic material or segments of DNA. So the function of crossing over is to produce new combinations of genes in a chromosome. So genes on a paternal chromosome will combine with genes from a maternal chromosome, and the point where the chromatids cross over is called chiasma. Now, at the end of prophase I, 3 events occur that are similar to prophase of mitosis. Okay, So in meiosis, at the end of prophase I, the nuclear membrane here disappears and the nucleolus also disappears. Then these two pairs of centrioles move to opposite poles of the cell, and spindle fibers are formed between the two pairs of centrioles. All this also occurs in mitosis. The next phase after prophase is metaphase I. In this phase, the homologous chromosomes are arranged side by side at the equatorial plane, or in the old syllabus of KSSM, we call it the metaphase plate. Alright, for KSSM syllabus, we use equatorial plane. So equatorial plane is an imaginary line in the center of the cell between the two poles here. Alright, where the homologous chromosomes will be arranged side by side. So this is a unique situation that we don't see in mitosis, because in mitosis the chromosomes are all arranged like in a single file like that. Here the chromosomes are paired up. That only happens in metaphase I of meiosis. So each chromosome, one chromosome of each pair of the homologous chromosomes, like this one, is attached to spindle fibers from one pole, while it's homolog, meaning the other member of the pair of homologous chromosomes, the other copy of the pair of homologous chromosomes, is attached to spindle fibers from the opposite pole here. That is to pull them and separate them, pull them apart and separate them so that they move to opposite poles later on. Okay? Now the sister chromatids are still joined or attached together at the centromere. The centromere will not divide in this meiosis I. That means in anaphase I it will not divide. Okay, so the important fact that you have to know here is homologous chromosomes are arranged side by side at the equatorial plane in metaphase I. Okay, memorize this because they like to ask what happens in metaphase I, metaphase II, and so forth. After metaphase I comes anaphase I. What is the important event in anaphase I? It's number 1 here. The spindle fibers contract and cause the homologous chromosomes in each pair to separate from each other and to be pulled to the opposite poles of the cell. So here we can see the pair of homologous chromosomes. When the spindle fibers contract, each chromosome will be pulled away from the other one, and they will separate and be pulled to the opposite poles of the cell. Okay, so the main thing that happens here is in anaphase I, the homologous chromosomes will separate when the spindle fibers contract and they are pulled to opposite poles of the cell. Then each chromosome still consists of 2 sister chromatids. If you look at this one, so you can see like 2 arms and 2 legs like that. You have to observe the picture carefully in order to find out whether it's anaphase I or anaphase II. In anaphase I, we can still see the sister chromatids attached together. So 2 sister chromatids are together and they're still attached together at the centromere, so they will move as one unit. Alright, so the centromere does not divide in anaphase I, and the sister chromatids are still attached together. Only the homologous chromosomes separate from each other in anaphase I, and they are pulled to opposite poles. Next, the cell goes into telophase I. In this phase, the chromosomes arrive at the opposite poles of the cell here. So each pole, for example this pole, now has a haploid number of chromosomes, or only one set of chromosomes. So one copy of the long chromosome, one copy of the short chromosome. That's one set of chromosomes. No more 2 copies of the long chromosome or 2 copies of the short chromosome. Now next, the three events that occur here, number 3, 4, and 5, also occur in mitosis. And it's the reverse of prophase. Now the spindle fibers disappear. Okay? Instead of appearing in prophase, now the spindle fibers disappear. The nucleolus reappear, again, the black dot, and the nuclear membrane is formed back again. Not shown here, but the nuclear membrane will be formed back again. At the same time, cytokinesis or division of cytoplasm is occurring, and we see the plasma membrane constricting here, or the cell constricting here, and it's going to divide into two, to form two daughter cells. Now what happens after telophase I? Cytokinesis or the division of the cytoplasm will occur, followed by interphase, and then meiosis II will start. So number 1, telophase I is followed by cytokinesis that produces two daughter cells. So here we see the process of cytokinesis starting to occur. The cytoplasm here, the cell here is constricting. Cell is constricting, and the cleavage furrow is forming. So this is an animal cell because of the formation of cleavage furrow. And then slowly the cell will split into two daughter cells. Both daughter cells produced are haploid, in a haploid condition, meaning that they have, each one has only one set of chromosomes, and only one copy of each type of chromosome. One long chromosome, one short chromosome. So each daughter cell receives one chromosome from a homologous pair. So for the long chromosome, this daughter cell has one copy of the long chromosome, and then this daughter cell has one copy of the long chromosome too. Same goes for the short chromosome. One short chromosome here in this cell, and one short chromosome in the other cell. So the interphase for meiosis I is usually short, and DNA does not replicate during this interphase. Now the events that occur in meiosis II after that are similar to those of mitosis, which we have studied earlier, except that in meiosis II, a haploid cell is involved. So whenever you see haploid cell undergoing nuclear division, we know that it's meiosis II. Whereas in mitosis, a diploid cell is involved. If you see a diploid cell undergoing the nuclear division, it's either mitosis or meiosis I. So if it's a haploid cell, it must be meiosis II. In meiosis I, cytokinesis occurs, and that is cytoplasmic division, followed by a short time of interphase. Then the cell goes into meiosis II, the second nuclear division. So in meiosis II, there are 4 phases, that is prophase II, metaphase II, ANAPHASE II, and TELOPHASE II. Or we can call it PMATI. Let's start with prophase II. Prophase II is the first phase here in meiosis II. The events that occur in prophase II are quite similar to the events that occur in prophase I of meiosis, except that there is no crossing over and no synapses. No synapses and crossing over. So let's look at the events that occur. The nucleolus and nuclear membrane disappear. Okay, so the nuclear membrane here is disappearing, and the black dot, the nucleolus, also disappears. Spindle fibers start to form in both daughter cells, and it forms in between the centrioles. So the spindle fibers will take part in the nuclear division later on to separate the sister chromatids. Centrioles move to opposite poles of the cell. Each chromosome is made up of 2 sister chromatids joined at the centromere. So this centromere has not divided yet, and the 2 sister chromatids are still attached to one another at the centromere. The daughter cell is a haploid cell. That means this cell has only one set of chromosomes of different sizes carrying different types of genes. Or we say that there's only one copy of each type of chromosome. One copy of the long chromosome and one copy of the short chromosome. Prophase II is then followed by metaphase II. In metaphase II, the main event that occurs is number 1. Chromosomes are arranged randomly on the equatorial plane here in each haploid daughter cell. So take note that the daughter cell is a haploid cell with only one copy of each type of chromosome. Now the chromosomes are arranged in the middle of the cell on the equatorial plane. Each chromatid is attached to the spindle fibers from both centrioles. So these spindle fibers are getting ready to contract in the next phase, right? Metaphase ends when the centromere separates or divides. Okay? So in ANAPHASE II, the centromere that attaches the sister chromatids together separate or divide. Then the sister chromatids in each chromosome separate as they are pulled by the spindle fibers that contract. So they separate and they are pulled to the opposite poles of the cell, led by the centromere. The centromere should go first because the centromere is the part that is attached to the spindle fibers. So when you have to draw this diagram, please show the centromere pointing towards the centrioles or the poles. Same for this one. Now each chromatid at this stage is known as a chromosome. So each chromatid will become a new chromosome for the new cell. After ANAPHASE II comes telophase II. So in telophase II, chromosomes arrive at the opposite poles, the two poles of the cell. Spindle fibers disappear as they have already done the job of pulling the sister chromatids to the poles. The nuclear membrane and nucleoli are reconstructed or they reform or they are formed around the chromosomes. The number of chromosomes of each daughter cell is half the number of chromosomes in the parent cell. So each chromatid will become a new chromosome, and we can see that now there's one long chromosome and one short chromosome at each pole and in each daughter cell. So there are only 2 chromosomes in each cell as compared to 4 chromosomes in the parent cell at the beginning of meiosis. So telophase II ends with the process of cytokinesis, the second cytoplasmic division, and it produces 4 genetically different haploid daughter cells. Okay, so for this intermediate daughter cell here, when cytokinesis occurs, it will form 2 haploid daughter cells, where there is only one copy of each type of chromosome. So, but there's one more daughter cell, which we did not show here, that divides to form 2 more haploid daughter cells. So that becomes 4 haploid daughter cells altogether, formed from one parent cell. So each haploid daughter cell contains half the number of chromosomes of the parent cell, and the genetic content of the daughter cells are also different from the diploid parent cell as they have undergone crossing over. And the haploid cells will develop into gametes. Lastly, here's a comparison of meiosis I and II, and this can be asked as an essay or structured question. Compare and contrast meiosis I and meiosis II. Now in meiosis I, synapsis, or the pairing up of homologous chromosomes, occurs in prophase I, but synapsis does not occur in meiosis II. That is, it does not occur in prophase II or even other phases. In meiosis I, crossing over or exchange of genetic material between the non-identical chromatids of homologous chromosomes occurs in prophase I, but in meiosis II, crossing over does not occur in prophase II. Thirdly, separation of homologous chromosomes which move to opposite poles occurs in ANAPHASE I of meiosis I, but for meiosis II, separation of sister chromatids occur, and the sister chromatids move to opposite poles. This occurs in ANAPHASE II. Lastly, at the end of meiosis I, 2 haploid daughter cells are formed in telophase I, but in meiosis II, 4 haploid daughter cells are formed in telophase II. That's the end of our lesson today. Thank you for watching. Please share, like, and subscribe. See you then. In the next lesson, we will discuss the differences between mitosis and meiosis for essay question. Goodbye for now.